Welcome everyone to this second session of contributed talks at EuroBioC 2020. Just a note before we start, as before, if you have a question, please put it in the Q&A tab in the right-hand side of your screen. And please, since we have multiple speakers in this session, don't forget to indicate who the question is for. So we ask it to the right person. So you can also upvote other questions if there's something you find particularly interesting. And with that, without further ado, I would give the word to our first speaker, who is Pierre-Luc Germain.
Thank you, Charlotte. So I'll speak about the SCDBL Finder package. I'll start with a brief intro on doublets for those who might not be familiar, and then I'll give a rough idea of the package and show you why it's a must for your single-cell sequencing pipeline. So a doublet is two cells captured in a single reaction volume, for instance, a single droplet, and that therefore generate a single transcriptomic profile. Now, when this happens with cells of the same type, we call them homotypic doublets, and for many purposes, these are relatively innocuous. Heterotypic or neotypic doublets are instead between cells of different types, and these can be very problematic. They can form their own clusters and appear as spurious new cell types. They can disrupt trajectory analysis and co-expression analysis. There are experimental approaches to deal with doublets, such as cell hashing or multiplexing samples of different genetic backgrounds. Cells that harbor multiple conflicting signals, like SNPs from different individuals, can thus be flagged as doublets. However, there are limitations to these approaches. They are not applicable in all circumstances, and even when they are, they cannot identify intra-individual doublets. And for these reasons, even with these approaches, we need further doublet detection. Now, what these technologies did enable us to do is first, they provide some positive doublets in real data. This confirmed, for instance, that the rate of doublet formation can be roughly predicted from the amount of cells loaded. And for instance, it's often as high as 15-20% in 10x data. In addition, such data can be used to evaluate doublet detection methods, and I'll do that a little bit later. There are already a number of doublet detection methods out there. I won't go in detail about the different strategies, but in short, our package essentially tries to take the best of all these different approaches. So the SCDBL Finder package includes a set of alternative tools and methods regarding doublets. It includes, for instance, those methods that were formerly in the SCRAN package and have been a little bit improved, but especially it includes a new eponymic comprehensive method, which is the one I'll be focusing today. So the method is very easy to use. It's a one-liner. What happens in a nutshell is this: after pre-processing, we generate inter-cluster artificial doublets. Of course, the devil is in the detail here. I won't get into that. We build a KNN network, assemble a set of statistics based on the network and also based on other properties of the cell. And these are going to include, for instance, the co-expression-based features. And then we train an iterative classifier to predict, based on these features, which cell is a real cell and which is an artificial doublet. Now, we carefully selected the features in order to avoid overfitting, because the problem in which we train is not exactly the problem that we are trying to solve. And also because the real cells contain doublets that are wrongly labeled as singlets, we do the classification iteratively. And at each round, we have a thresholding procedure that is used to turn the score into a binary call. The thresholding is not very complicated. It's essentially trying to minimize the misclassification rate of artificial doublets while also minimizing the deviation from the expected doublet rate. And since the expected doublet rate can also be given an uncertainty range, the user can decide to put more or less weight on the misclassification rate if, for instance, the user doesn't know even roughly what the expected doublet rate is. Okay, so all right, if you have multiple samples, which are here understood as separate captures, what we do is that we split by sample to generate the artificial doublets and the KNN. And this not only speeds up the process, but it also makes it more robust to sample-specific or batch effects. And then we train the classifier on the whole data set and split again for the thresholding, thereby accounting for eventual differences in doublet rates between captures. Okay, this being over, I now turn to the question of how well SCDBL Finder performs. There's a really nice preprint benchmark out there in which the authors assembled many datasets with known doublets in order to evaluate published doublet detection methods. The big limitation here is that only part of the doublets are flagged, and some that are flagged are homotypic and thereby virtually undetectable. But to cut a long story short, the upshot was that on pretty much as aspect they looked at, the top method was this doublet finder package. Unfortunately, the benchmark did not include my package, but since they made everything available, I could run their benchmark and compare SCDBL Finder to this top alternative method. And this is what you see in those plots. So you see the area on the ROC curve and on the precision and recall curve for SCDBL Finder and Doublet Finder on each axis. And each dot is a data set. So in both panels, you can see that SCDBL Finder is superior in the majority of data sets, sometimes by a lot. And when it's not, it's very close to the diagonal. In addition, SCDBL Finder runs about 10 times faster, and these are all small data sets. It actually scales also better. So in other words, it offers better accuracy than the best alternative and much faster. This is chiefly because we built on a number of bioconductor resources to optimize every step. So it's also a lot due to the work of other people. Okay, finally, an added value of our method is that we can tell you where the doublets came from, and therefore can also tell whether a data set shows enrichment for specific types of doublets, which can take the form either of really enrichment for a specific combination or enrichment in a given cell type forming doublets with anything else. So we formalized these two problems into tests, which we are currently trying to validate. It works really well on simulation, but we are trying to see how it works on real expected biological expectations, and these will be available soon. Okay, so in summary, the take-home messages are that first, you should be careful about heterotypic doublets, and that fortunately we can identify them pretty accurately, and that the SCDBL Finder method is very easy to use, outperforms alternative methods, runs considerably faster, scales better, and offers more functionalities. So we invite you to try it. Don't hesitate to contact us if you have questions. This is under constant improvement, so please use a recent version. And with this, I'd like to thank the Mark and the Robinson group, especially Will McNair, with whom I had a lot of doublet-related discussions, and also Aaron, who contributed especially the Scrum-based methods, but also helped with a lot of little technical things. And thank you all for your attention.
Thank you very much. So we have one question here in the Q&A from Kevin, who's wondering if there is a sense of how many, or actually how few doublets are needed to disrupt an analysis, for example, a trajectory analysis, either in terms of the number of doublets or in the proportion of the full data set.
That's a good question. I wish I had expected it and had a good answer. The benchmark paper that I mentioned actually looked a little bit at this, but didn't specifically ask the amount, but it tried to study the impact on simulated trajectory, for instance, and also on simulated differential expression settings. And I mean, it does clearly have an impact, but how big it is and how many doublets you need in order to have an impact, I wouldn't be able to say. But that's definitely something to investigate. Thanks. Are
there any other questions? Otherwise, I have a question. So was this mainly tried on 10x genomics data or also other droplet platforms?
Other platforms as well. Although I must say most of the data sets that I've been using have been 10x, but it's been part of the benchmark includes some other data sets as well. The one thing I would say, I've tried the method also on single cell ATAC-seq data, and there you need to increase the number of features that are used. Otherwise, it sort of works, but I don't think the parameters are optimal at the moment. So this is something that will require further work. Thank you.
Okay, and the slides are posted in the chat. Yeah, so we have one more question, which is whether doublets are also a problem for plate-based protocols.
I mean, I guess it depends which plate-based protocol. I think the Fluid IMC one had this step back in the days, at least, where there were images being taken, and this was used in QC to determine whether you sequence a given well or not. So in this case, I guess you can avoid it, but I wouldn't be able to say about recent technologies. I haven't worked with plate-based technologies in a while.
So I think in the interest of time, I will thank you very much there, and we will move on to our second speaker, who is Andrea Sottosanti.
Good afternoon, everyone. Thank you to be here. Before starting, I want to tell you that this is an ongoing project, so every suggestion is very welcome from you. And my research work is basically on the 10x genomics dataset. So this technology is a new sequential technology which allows to have not just the expression of really thousands of genes, but also a kind of reconstruction of the spatial structure of a tissue, of a sample of tissue. So for example, here, starting from this slice of the tissue, you can get, using these spots, you can get the expression of thousands of genes inside each spot, which more or less corresponds to a single cell. Every spot more or less corresponds to a single cell. So in literature, this kind of new technology has increased the interest for the so-called spatially expressed genes. So these are genes for which the expression in a cell is related to the expression in the surrounding cells. But the idea is that even though there are methods in order to detect these so-called spatially expressed genes, there are still some things that must be investigated. So first of all, these methods do not account for the presence of multiple types of cells. And second of all, some genes or cluster of genes might be spatially expressed just in some specific cell types. So we want to put all these things into a statistical model in order to evaluate these kind of aspects. So we start from a data matrix, an n times p, where n is the number of genes and p is the number of cells, so that the element ij is a measure of the expression of the gene I into the spot j. So what we have is that we have the spatial location of the spots, S1, SP, and also right now we are just assuming that there is only one type of cells at the base of the data. So the idea is that there are two types of correlation. The first one is the correlation among the genes, and so among the rows of the data matrix, and the second one is the correlation of the spots, so the columns, which might be related to the spatial structure of the data that we have. So we want to establish a statistical model over the entire data set. We use this so-called matrix variate normal distribution, which is basically a distribution over the entire data matrix. So it is parameterized by a mean matrix and two covariance matrices. The first one for the rows and the second ones for the columns. So the idea, the base of this model is that every row, every column, and every element, and even every block of the data matrix is Gaussian distributed. And so now let's characterize these kind of elements. Regarding the rows, so the correlation of the genes, we assume that we have a diagonal matrix. But the idea is that because we want to include the correlation among the genes, we specify a kind of mixed effect model by assuming that each and every variance of the genes is distributed as an inverse Gaussian model. So the idea is that because of this matrix is very big, but actually can be sparse matrix, in order to avoid the estimation of all the elements, we induce this mixed effect model in order to account for the correlation of the genes. In the same way, we specify the correlation of the cells using this structure. So for those who are familiar with Gaussian processes, this is a spatial kernel matrix, which is a matrix that depends from the distances among the spots. And the idea is that this tau parameter is positive and represents the spatial expression that we have inside the data, while this psi is a nugget effect, so a part of the variability which cannot be imputable to the spatial structure. Okay. But as I told you before, we don't have just a single type of cells. We have multiple types of cells, and also we might have even a cluster of genes. So the idea is that our data matrix can be split into blocks rectangular blocks, which are even called co-clusters in the statistical literature. And each and every block, we assume that is matrix variate normal distributed, and with its own parameters, and even with its own spatial parameter and nugget effect. So by doing so, we are allowing every block which represents a group, a cluster of genes, which has a kind of spatial expression in a certain type of cells. This kind of model can be estimated using an EM type algorithm. We are still working on the estimation algorithm, so now I will give you just some preliminary results. But we exploited the human dorsolateral prefrontal cortex spatial data generated by the 10x Visium technology, and which is collected by the R package spatialLIBD. We reduce the data set in order to have the most variable genes, the first 1,000 most variable genes measured in 1,585 spots. And we run our model considering just a single cluster of genes, but 4 different clusters of cells. So these are the results. The one on the left are the clusters reported by the spatialLIBD package. The one on the right are the one that we detect with our model. So as you can see here, we distinguish like 2 macro areas. The first one is within these boundary cells, which are blue and orange, so the part within these spots, while the second one is outside. And for each and every cluster, we distinguish, we have the parameter estimates, so the average expression, as well as the ratio between the spatial parameter and the nugget effect, which tells us the amount of spatial expression with respect to the nugget effect in each and every cell type that we distinguish. And in the same way, we can have we can look at the expected variance for each and every gene within the cell types. And it's a kind of posterior distribution, this one. And you see here in particular that the variation of the genes now accounts also for the spatial expression. So you see here that the variation changes with respect to the cluster of cells that we identify. So for example, in the cluster 3 here, that corresponds to the dark green points, has the large variance for the first 2 genes was called these ones. Okay, and these genes are mostly related with the oligodendrocytes cells. So these are the preliminary results that we have from our model. Any kind of suggestion is really very welcome, and so feel free to contact me, and thank you for your attention.
Thank you very much, Andrea. While people think about their questions, I had a question about the pre-processing, I guess. So you assume a normal distribution and you feed in log transformed counts.
Yeah,
did you
exactly.
try other types of pre-processing, other types of transformations?
No, not yet. We are working on this part.
Okay. Are there any other questions? So I have another thought. So I'm kind of so if you have like the same cluster of cells or the same group of cells in 2 very distinct parts of the of your tissue. So would those, like, kind of which dominates, like the gene expression or the spatial distribution? Would those be classified as the same into the same cluster or in different clusters?
I mean, I think that if I correctly got your question, I think it's like the model accounts for this by estimating the scale of the spatial structure very high. Mm-hmm. So there's a scale also for the spatial structure that is estimated together with the other parameters. So if you have 2 like coherent cluster of cells, you will have also a very large estimate of the scale parameter. So they will become as a unique cluster. I don't know if I correctly, if I actually answered your question.
Yes, thank you. So I think maybe if you have questions for Andrea later, feel free to reach out during in the platform. And I think we'll move to the next talk, which is by Kwame Forbes.
Hello everyone, thanks for having me. My name is Kwame Forbes. I'm a postbac researcher at the University of North Carolina at Chapel Hill, conducting computational biology research. I've always been interested in how the use of computer softwares can be used to answer very complex biological questions that would be impossible for humans because of the high volume of data. Currently, I'm working with Dr. Michael Love on developing new functions within the DC2 software for the analysis of RNA datasets. With these new functions, we hope to help biologists analyze and interpret their bulk datasets with the integration of a single-cell dataset. This would allow biologists to know which cell types may be of importance when they see differences in RNA in their samples. With this in mind, we are in the process of developing a simple interface that will allow users with bulk RNA-seq differential expression results to use publicly available single-cell datasets by visualizing the cell type-specific expression patterns of their genes of interest. So to accomplish this, we develop a function called integrate with single cell within DC2, which takes two arguments, and based on whether your bulk dataset is either human or mouse, would generate a list of corresponding single-cell datasets. So in our workflow, we selected 8, which is the PBMC 10x dataset. So currently, our integration only allows for human or mouse, but as more single-cell datasets are allowed in bioconductor, we plan to expand into other organisms as well. So moving along, the function's output will be a list that contains the original results table, the bulk gene count, and the single-cell object selected by the user. So next, to perform unbiased cell type recognition, we use the package on bioconductor called single-R. So although single-R is used in our workflow, ultimately we decide to leave the user the choice of a means of cell type recognition. Along with the integration of a single-cell dataset, a great way to help biologists further analyze and interpret their bulk dataset with the integration of a single-cell dataset would be to provide visualizations in the form of plots or graphs. So this led me and Mike to in the process of creating a new package called Viz with SCE. So currently this package takes two arguments, the results from the integrate with single cell and a second argument called which. So the which argument is the gene used for that will be used for the viral impact, and it's represented as a number that corresponds to the gene. So with the n smallest adjusted p-value. So for instance, 1 for the gene with the lowest adjusted p-value, 2 for the gene with the second lowest adjusted p-value, and so on and so on. So looking at figure 2, the argument which equals 2, showing the gene DUSP1, and for figure 3, the argument of which is which equals 3, showing the gene SAMHD1. And we could see that in both genes, monocytes are highly regulated and expressed. So the next thing that me and Mike wanted to do was make the visualizations more interactive, and to accomplish this we use the package also on the package called Shiny because of its popularity and simplicity. So here we were able to create a very basic interactive violin plot, and we decided to use a slider button so the user in real time could select their gene that they want to analyze and interpret. So to conclude, this new function integrate with single cell, we were able to get it into the new release of DC2. And right now we are in the process of showing the demos to many PIs, to some PIs at the University of North Carolina at Chapel Hill. And in the future, along with adding various other plots and other statistical tests, we are interested in exploring launching with IC because it's a well-developed package for the integration and exploration of single-cell datasets. And with that, I would like to thank you all for listening to my presentation, and I would leave a link in the feed where you can provide some feedback for the overall project. Thank you.
Thank you very much. We have time for questions. Maybe I'll start. So you said it can use human and mouse data, and it seemed to identify the species automatically. So how does it do that? What information does it use for that?
Thank you for the question. Well, because we are using the datasets found on Bioconductor, they are well organized based on their species. So, for example, all single-cell human datasets have a distinct pattern that our code looks for, and likewise for mouse, if that answers your question.
So it also identifies the species from the bulk results from the DSeq dataset?
Yes.
Okay. So we have a question from Davide, who says, do you think it's possible to apply your method to find clusters of genes, I think, starting from some genes of interest?
Well, right now, since we are in the beginning stage of the research, we haven't really given it much thought, but in the future, as we continue to see all that we're capable of, it would be possible to see if it's possible.
I guess this is a question for you. So did you have a look at PDX types of data sets?
No, we have not right now.
Yeah, I was also interested in I had a question about the multi-gene. So what kind of multi-gene, what kind of gene groups would you imagine that people would put in? Would it be like the top regulated genes, or would it be like a gene set of a specific function, or what kind of applications do you envision there?
Well, right now I envision, like you said previously, like the top genes, and yeah.
Okay, great, thank you so much. And I think we will go over to our last talk of this session, which is Brenda Pardo.
So good afternoon. My name is Brenda Pardo, and I'm an undergrad student in genomic sciences at Universidad Nacional Autónoma de México, and I'm currently performing a research internship at Leonardo Collado Torres' team at Libre Institute for Brain Development. And today I'll talk about my work on adapting the package spatialLIBD to use our objects structures specifically to store spatial transcriptomics data. Okay, so Maynard Collado Torres et al. Used the 10x Genomics Visium platform to define the spatial topography of gene expression in the 6-layer human dorsolateral prefrontal cortex, DLPFC. And they identified layer-enriched genes. They overlaid the laminar expression signatures onto single nucleus RNA sequencing data. And they showed the differential layer-enriched expression of genes associated with brain disorders, just as schizophrenia and autism spectrum disorder. And to interactively inspect the DLPFC spatial transcriptomics data, they created a bioconductor package, spatialLIBD. And this package contains functions for accessing the spatial transcriptomics data, also for visualizing the spot level spatial gene expression and clusters, and to inspect the data interactively, either on the user's computer or through a web application. And here I'm showing you a screenshot of the web application. So the spatialLIBD package used to employ our objects from the single-cell experiment class to store the data Nevertheless, Righelli and Rizzo created a more accurate class called VisiumExperiment for spatial transcriptomics data. So Visium inherits slots and methods from single-cell experiment and from spatial experiment, however, allows an optimal accommodation of the spatial transcriptomics data. And it contains specific slots to store the spatial coordinates for its small group of cells containing a spot. Also, the paths for the tissue images, information about which spots are covered by tissue, and the scale factors to know the location of the spots in pixels, and methods to set and retrieve this information. The class was designed to hold a unique sample, and it is contained in the package spatialExperiment. So we perform modifications to the set of functions that make up spatialLIBD so that they could work with VisiumExperiment objects. First, our function fetch data that was previously designed to retrieve a single-cell experiment object containing the DLPFC data. With our updates, the function is able to return an object belonging to the VisiumExperiment class. And it is important to say that our VisiumExperiment object is a little bit more complex than a regular VisiumExperiment one, because it contains multiple samples, and this has an impact on the arrangement we made for part of the data. For example, we accommodated data into our VisiumExperiment object to contain scale factors for multiple DLPFC tissue images, and also to contain local image paths for these tissue images. Next, we adapted the function for visualizing the histology image from Visium. This function is called geomSpatial to use VisiumExperiment objects. And lastly, we modified our functions to visualize the gene expression and clusters for a given sample at the spot level to use objects from this class. And some of the challenges we faced when using VisiumExperiment class to contain our data were that first, VisiumExperiment class validity code checks that the image files exist locally instead of being available remotely through an URL. And because our tissue images are in an URL, we decided to download the images at the moment the VisiumExperiment object is created. And second, the scale factor slot, as it is designed to handle a single sample, has a storage limitation. It has to contain a list with the exact names defined by Righelli and Rizzo for the 4 scale factors, which are the ones indicated here. And so, given that we don't have just one but multiple samples, we decided to create a list with the required 4 scale factors names, but also with a 5th element of the list, with the full list of scale factors for all of our samples. And also, we created a function called update scale factors that updates the scale factors for a given sample ID, putting the sample values in the first 4 elements of the list, which have the names defined by the authors of the VisiumExperiment class. And here, I am showing you an example of one of our spatialLIBD functions working with an VisiumExperiment object. So we retrieve here a VisiumExperiment object containing DLPFC data. Then we generate an object containing data for just one sample, and we use this function called SCEImageGene in the data to visualize the expression for a given gene at the spot level. And here I am showing you the generated image where we are observing the log counts for a gene for each spot. The future plans for spatialLIBD are based on work with Helena L. Crowell, Lucas Weber, and Dario Righelli, and are to decide if it is optimal to move the package graphical functions to a new package, also to update spatialLIBD based on the new version of spatialExperiment. And in addition, we will write an application note about the package. And well, my personal plans include to learn more about data analysis, working with 10x genomic data with Leo and colleagues, colleagues at LIBD while potentially working on more R packages, as I keep learning new skills that will help me join grad school programs. And if you want to know more details of this work, I invite you to read our blog post on LIBD R Stats Club blog. And finally, I just want to acknowledge all the people involved in this work, in the project of transcriptome scale spatial gene expression of the LPFC, and in spatialLIBD from Liver Institute and from Johns Hopkins Biostatistics Department. And especially thanks to my mentor, Leo. Thank you.
Thank you very much. So we have time for some questions. I have maybe one question. So you put the objects contain the paths to the image. So how do you suggest to share this object? If you want to share an object with someone, do you need to update the path, or can it be like I don't know, automatically updated? You need to remember to share the images as well separately. So how would you suggest that to be done?
Yeah, so we are thinking about we want that the object allows to include not local paths, but URLs. So in that way, it's easier to share the information, and you just download the images whenever it is necessary, and just the subgroup of images that you need, instead of downloading everything at the beginning, and it's more difficult to share.
Yeah. So we have one more question from Friederike, who was asking, you mentioned that this was for a single sample. How would you go about with multiple samples?
Okay, yeah. So Elena and Dario, we met, and they are working on make this VisiumExperiment object for multiple samples, including these difficulties with having multiple scale factors. So yeah, they are planning to make this object available to include multiple samples.
Work in progress. Okay, thank you very much. I would like to thank all the speakers of this session again for your great talks. And just to say that there were a couple of questions that came up later for the earlier speakers. So if your question was not answered, feel free to reach out to the speaker in the platform or outside during the week. So thank you again. And now we have a 10-minute break before the next session of contributed talks continue at 5 past. So thank you very much.